An attractive young woman gains global media attention for developing an innovative, potentially world-changing scientific technique. The female scientist seems to have a way with older men in positions of authority, who praise her as a genius and visionary. But her work is eventually revealed to be fraudulent, and her scientific technique doesn't actually work. A senior scientist on her team commits suicide, and her older male supporters see their reputations tarnished. While this could describe the story of Theranos founder Elizabeth Holmes, it's actually the story of a Harvard-trained Japanese scientist called Haruko Obokata. In 2014, just 1 year before the Theranos scandal was exposed, Haruko Obokata's rise and fall was featured in publications such as The Wall Street Journal, The New Yorker, and The Guardian. But her story has since fallen into obscurity, probably because her downfall was as swift as her rise, and afterwards, she vanished from public view. In 2014, at just 30 years old, Obokata claimed to have discovered a new method for generating stem cells from ordinary cells, such as skin cells. Her idea was simple. Placing ordinary cells in an acid bath for approximately 30 minutes would trigger stem cell-like abilities in the few cells that survived the harsh conditions. The surviving cells would recover in as little as 3 days, reprogrammed into a stem-like state. Stem cells are undifferentiated cells with the ability to develop into various cell types. Adults have stem cells in some tissues, but they possess limited potential and can only create cells in their native tissue. In contrast, stem cells from embryos have the capacity to become any cell type in the body. Such cells are known as pluripotent. The emergence of stem cell science presented a revolutionary opportunity for patient-specific tissue growth that could be used for organ or tissue transplants, without concerns of immune system rejection. But the use of human embryos in stem cell research was met with moral and ethical controversy, particularly among those who believed that life began at conception. Haruko Obokata's technique, which transformed mature cells into pluripotent stem cells, promised to circumvent these ethical dilemmas. She dubbed her method, stimulus-triggered acquisition of pluripotency, or STAP, and introduced the technique in 2 groundbreaking papers published in the prestigious journal Nature. Just 2 years earlier, a fellow Japanese scientist, Shinya Yamanaka, had won the Nobel Prize for a different technique to convert mature cells into stem cells. However, his technique, which relied on genetic manipulation, was costly, unreliable, and the cells it produced were potentially cancerous. Obokata's discovery threatened to make Yamanaka's approach obsolete, sparking legitimate Nobel Prize buzz around Obokata herself. The media focus on Obokata became extreme. She became a scientific hero in Japan. But the media's interest went far beyond her scientific work. They wanted to know what she wore, how she decorated her lab, and what she did in her spare time. This frustrated some Japanese women, who saw the coverage as misogynistic. The focus on Obokata also frustrated some of her male colleagues. Obokata was the first author on both articles, but she had co-authored the papers with several male scientists, and had built on the previous work. Some of them felt their contributions to a potentially Nobel-worthy discovery were being ignored. However, Obokata's research and reputation began to unravel within weeks, and her papers were retracted within 6 months, partially thanks to the power of the Internet, where eagle-eyed readers around the world began to identify problems with her articles. Meanwhile, other labs were struggling to replicate her work. Obokata was eventually found guilty of scientific misconduct. Yet during and after her downfall, the Japanese media's obsession with her continued. Paparazzi pursued her relentlessly. A popular video game created a character based on her. Her scientific career in shambles, humiliating rumors circulated in the press that she would pose nude, out of desperation for cash. The fixation persisted until she effectively went into hiding. In the end, Obokata and one of her male co-authors pointed fingers at each other for the fraud. Official investigations could not pinpoint exactly who was responsible. But Obokata had received all the attention on the way up, and she received all the attention on the way down. Additionally, she was exposed for a series of lies and deceptions that did not help her case. Haruko Obokata's story has several clear parallels to that of Elizabeth Holmes. They both demonstrate how the media and public can build up a young attractive woman who manages to succeed in a male-dominated field, and how this same media and public can also excessively revel in a young woman's downfall. The story of STAP cells begins with a renowned Harvard scientist, Charles Vacanti. A tissue engineer best known for the ear mouse, Vacanti made headlines by implanting an ear-shaped scaffold seeded with cow cells under the skin of a living mouse with a very weak immune system. This allowed a biodegradable, cartilaginous ear to be grown from donor cells without immune system rejection. His pioneering work continued with the engineering of a trachea for a 14-year-old girl with a deadly tumor. Vacanti was initially trained as an anesthesiologist but had ventured into a new field of tissue engineering so successfully that he eventually opened a tissue engineering lab at Harvard. In the late 1990s, Vacanti developed an interest in yet another field, the emerging field of stem cells. Vacanti partnered with one of his brothers, Martin, to delve into stem cell research. One day, Martin, who had been struggling to isolate neural stem cells using conventional methods of tissue agitation, finally managed to isolate curious-looking cellular specks by aggressively scraping the tissue with a scalpel. But when he took his specks to an expert, they were dismissed as junk. Discouraged, Martin placed the specks in an incubator, planning to dispose of them properly after the weekend. But when he returned on Monday, he was surprised to see that the specks had given rise to cellular spheres, as would have been expected from stem cells. Intrigued by this unexpected result, Charles Vacanti theorized that the aggressive scalpel scraping might have induced stem cell formation rather than merely isolating stem cells from the tissue. Charles and Martin referred to these small structures as spore-like cells. He proposed in a research paper that such cells lay dormant in all tissue types, and possessed the ability to regenerate tissue lost due to injury. The theory was dismissed by other scientists as nonsense. At one conference, Charles Vacanti was heckled and told to stick to tissue engineering. Despite the skepticism, Vacanti remained confident in his discovery, and sought a researcher to assist with further investigation into the spore-like cells. A Japanese colleague recommended one of his students, Haruko Obokata, who had graduated at the top of her class in chemical engineering at Waseda University. Upon joining the Harvard lab, Haruko Obokata quickly gained a reputation as a dedicated and diligent worker. Demonstrating exceptional skill in cultivating Vacanti's spore-like cells, she decided to pursue her PhD on the topic. She successfully induced the spore-like cells to produce teratomas, tumors that were considered evidence of pluripotency. Obokata also refined the method for growing spore-like cells. While Martin Vacanti had experimented with mechanical stress, freezing, starvation, and oxygen deprivation, Obokata devised a more standardized approach by exposing the cells to an acid bath. Scientists had developed a way to test cells for pluripotency by engineering them to glow a fluorescent green. Obokata's cells successfully exhibited the green glow. After earning her PhD, Obokata continued her investigation into spore-like cells at Japan's esteemed Riken Institute. Established in 1917, the nationally funded institute had produced 2 Nobel Prize winners in physics and had hosted other Nobel laureates in medicine and chemistry. Still only in her 20s, Obokata was granted her own lab at Riken's Center for Developmental Biology, or CDB. From there, she continued to collaborate with Vacanti's lab at Harvard on stem cell research. Vacanti and Obokata sought to improve the chances of getting published in Nature by collaborating with a high-profile co-author. Koji Kojima, Vacanti's lab director, connected them with his old friend Teruhiko Wakayama, a renowned scientist credited with being the first person to clone a mouse. Wakayama was intrigued and enthusiastically partnered with Vacanti and Obokata. He supplied Obokata with mouse cells, which she then exposed to acid to produce STAP cells. Obokata then returned the cells to Wakayama, who used them to grow a chimera, a mouse fetus developed from STAP cells injected into a mouse embryo. Wakayama personally created the chimeras and refused to share the process with Obokata, fearing she might abandon him if she could perform the procedure herself. Despite the collaboration with Wakayama, their initial submission to Nature was rejected. The editors felt there was insufficient evidence to prove the cells had been converted. They argued that the process might have simply isolated regular stem cells, or may somehow have been contaminated by them. The paper was also rejected by Cell and Science. To bolster their evidence, Obokata, Vacanti, and Wakayama decided to include additional steps demonstrating that the new cells were indeed converted somatic cells. Obokata identified a cellular marker unique to mature cells and revealed its presence in the converted stem-like cells. Wakayama devised another approach, using the STAP cells to cultivate a stem cell line. The ability to develop a line of infinitely replicating cells is a unique characteristic of embryonic stem cells not found in somatic cells. Wakayama claimed to have grown a stem cell line from some STAP cells provided by Obokata. This surprised Obokata. She had never been able to grow a stem cell line herself. Wakayama attributed his success to specialized methods. Yoshiki Sasai, one of the founders of Riken Center for Developmental Biology, had taken a keen interest in Haruko's work. A senior scientist known for successfully growing the optic cup and parts of the brain from stem cells, Sasai had published in prestigious journals like Nature and Cell. He believed he could present the research in a way that increased its likelihood of publication. He joined the paper as a co-author and was the person who coined the memorable acronym STAP. Other researchers at the lab grew cautious of Sasai's relationship with Obokata. They observed that Obokata tended to form close ties with men in positions of authority, while her relationships with her peers remained distant. They also perceived Sasai as being overly enchanted by Obokata, whom he called a genius. One Riken researcher accused Sasai of neglecting his own research to work on hers. Charles Vacanti also became concerned about Sasai and Obokata's relationship. When he reached out to Obokata for research updates, he would receive responses from Sasai, who claimed she was busy. Vacanti worried that Sasai was trying to exclude the original Harvard team members from the research, so that Obokata could take all the credit. Nonetheless, with Sasai's assistance and the incorporation of the additional verification tests, Nature ultimately accepted the research for publication. Haruko Obokata's co-authors included scientists from both the Harvard lab, including Charles Vacanti, his brother Martin, and his lab director Koji Kojima, and scientists from Riken, including Yoshiki Sasai and Teruhiko Wakayama. Following the unveiling of her research, Haruko Obokata was catapulted to stardom. At first, she gladly welcomed the limelight. In stark contrast to Elizabeth Holmes, Obokata crafted a distinctly feminine persona. Elizabeth sought to emulate a masculine image, donning a Steve Jobs-like turtleneck, speaking in a deeper voice, and claiming to live a solitary life dedicated to nothing but work. Elizabeth Holmes also claimed to live alone in a small apartment when she in fact lived in a $10 million mansion with her secret boyfriend. Haruko, on the other hand, embraced her femininity and portrayed herself as having a balanced, active social life. She revealed that she had a boyfriend, went on dates, loved shopping, had a pet turtle, and played lacrosse in college. Newspapers detailed her lab decorations, highlighting the pink doors, pink trash cans, and cheerful yellow walls adorned with Moomin's cartoon stickers, and her preference for wearing a traditional Japanese housewife apron, or kappogi, instead of a lab coat. As her fame grew, retailers noted a surge in kappogi sales. But the emphasis on her appearance irked some female Japanese viewers. On one television show, a popular TV personality introduced Obokata's discovery by saying, "A very cute, apron-wearing girl has just announced amazing research that could repaint the story of life sciences." Bloggers assailed the coverage of Haruko as misogynistic. Some people objected to the frequent use of the term, rikejo, meaning, science girl, to describe Haruko, arguing that it suggested women in science were somehow unusual or out of place. Meanwhile, Vacanti's unease about the Harvard team being overlooked intensified. Nature's press briefing on the discovery featured only Haruko and Sasai. Vacanti was taken aback when he saw a video on Nature's website, in which Haruko claimed to have single-handedly conceived the idea for STAP, claiming that the idea of stress-altered cells had come to her while taking a bath. She went on to claim that she had subjected the cells to various stressors, such as starvation and freezing, before finalizing her method. However, the initial idea for STAP had been Charles Vacanti's, and the experiments she mentioned, starving and freezing cells, had actually been carried out by Charles's brother, Martin. In essence, she had plagiarized their origin story. Upon watching the video, Martin Vacanti immediately called his brother in disbelief. Nevertheless, Vacanti continued to heap praise on Haruko, telling her that she deserved all the attention she was getting. He told her she was the most intelligent and hard-working scientist he had ever had working in his lab, and also the most beautiful. However, Obokata's honeymoon period with the press would soon end, in part due to the Internet's ability to make it faster and easier for a large number of individuals to access, analyze, and compare information. Both articles were posted on PubPeer, a website established in 2012 for people to share scientific papers and discuss them openly. Within 2 weeks, someone noticed that 2 images in one of her papers, an image claiming to depict a placenta created from STAP cells and another showing a placenta created using a different method, appeared to be identical. Obokata brushed off the error as a mix-up of image files on her computer. Soon after, someone observed that an image of PCR gels seemed to be manipulated, with one column spliced into the image in a way that made the data appear more persuasive. Obokata argued that she had merely rearranged the columns to clarify the data, not to deceive anyone. Yet, the issues only escalated. A blogger in Japan obtained a copy of Obokata's PhD thesis and discovered that images from her thesis resembled images from her STAP papers, despite the fact that her thesis had been on a different experiment, the growth of teratomas. Furthermore, it was revealed that large portions of her thesis were plagiarized directly from the NIH website. Obokata would eventually try to explain the plagiarism by claiming that she had accidentally submitted a draft of her thesis for binding, instead of the final version. By this stage, skepticism surrounding STAP had escalated dramatically. The paper's authors conceded that the papers should be temporarily retracted until the data could be re-examined. Vacanti asked Obokata point-blank if the data was trustworthy, and she assured him that she had not falsified any data. Meanwhile, laboratories worldwide were struggling to replicate Obokata's experiment. George Daly, the director of stem cell transplants at Boston Children's Hospital and head of his own lab at Harvard, sent one of his graduate students to Vacanti's lab to investigate. The student discovered that Vacanti's team had misinterpreted a crucial aspect of the experiment, the green glow. The glow observed in their STAP cells was not actually an indicator of pluripotency. Instead, it was autofluorescence, a natural emission from cells when they are dying. A simple test could have differentiated between the two phenomena. A green glow for pluripotency should have been visible only through a green filter, but the STAP cells glowed through both red and green filters. Daly's graduate student reported that Vacanti's team didn't know what they were doing. This was unsurprising considering that they were originally a tissue engineering lab, and were venturing into a new field. This meant that the STAP cells had never been pluripotent to begin with. Therefore, the follow-up experiments conducted by Obokata and Wakayama to confirm pluripotency, including the chimera and the stem cell line, must have been fabricated. George Daly proceeded to analyze the genomes of the STAP cells and found that they did not even match the mice from which they were supposedly derived. The original mice cells were female, while the STAP cells were male. Obokata claimed she didn't know how this was possible. There was only one plausible explanation. The so-called STAP cells were actually ordinary embryonic stem cells. At some point, someone had introduced embryonic stem cells into the experiment. There were only 2 ways this could have occurred. Either Obokata had given Wakayama embryonic cells instead of STAP cells, or Wakayama had used embryonic cells to create the chimera and stem cell line instead of the STAP cells Obokata had given him. Wakayama and Obokata pointed the finger at each other. Obokata claimed there was no way that she could fool an expert like Wakayama, and that the only place she got the cells in the first place was from Wakayama's lab. Wakayama insinuated that Obokata might have snuck embryonic stem cell into the lab. Police questioned Obokata on charges of theft after another Riken researcher filed a report that embryonic stem cells had gone missing from another lab at Riken during the time she had worked there. But nothing was proven. At Daly's lab at Harvard, scientists prepared a report debunking the STAP cells, while Japan's Riken Institute conducted its own official investigation. Both teams concluded that the experiment had likely been contaminated by embryonic stem cells, but neither could pinpoint the individual responsible. However, the media seemed to have made up their mind, directing the blame towards Obokata. A member of the review team at Harvard also privately admitted to a journalist that he too placed the blame on Obokata, saying that, clearly, what happened was that Obokata had given Wakayama standard embryonic cells, falsely claiming they were STAP cells, and that Wakayama had believed her. Wakayama, whose achievement of being the first to clone a mouse had established his scientific reputation and had been successfully replicated by other labs, had no history of scandal and was seemingly above suspicion. In contrast, Obokata's credibility was marred by doctored images, plagiarism, and shoddy record-keeping. In some cases, Riken investigators struggled to find evidence that some of her experiments had ever even been conducted. Nonetheless, they did attribute some responsibility for the scandal to her more experienced co-authors for their inadequate oversight of her work. As the scandal surrounding the STAP cells continued to unfold, Obokata's mentor at Riken, Yoshiki Sasai, became increasingly despondent. He had been one of Obokata's most vocal supporters, comparing her work to Copernicus's reorganization of the cosmos, and now the Center for Developmental Biology, which he had co-founded, faced potential closure or funding cuts from Japanese authorities as a result of the scandal. Those close to Sasai observed that he seemed to personally feel a disproportionately large amount of responsibility for the controversy. He spent a month in the hospital receiving treatment for depression. In August, a month after the STAP papers were officially retracted, a security guard discovered Sasai's lifeless body, hanging in the stairwell of the CDB Institute. He had left a note for Obokata, urging her to be sure to reproduce STAP cells. Obokata was deeply saddened by the loss of her mentor but chose not to attend his funeral, fearing that his wife and children blamed her for his death. The Riken Institute provided Obokata an opportunity to recreate her STAP experiment under strict supervision. For months, she worked in the lab, attempting to reproduce the STAP cells. Surveillance agents tied on her lab apron every day, and checked her pockets to ensure she didn't bring any external items. She wasn't allowed to handle reagents, or analyze her data independently. Obokata later expressed the feeling that she was treated like a criminal. She also believed that being prohibited from analyzing the data herself prevented her from knowing if the experiments were going well. After 6 months of unsuccessful attempts to recreate STAP cells, Obokata resigned from Riken, and eventually, Waseda University revoked her PhD. One might wonder why Obokata would choose to lie. Unlike Elizabeth Holmes, Obokata had actually graduated, at the top of her class no less, and was widely considered brilliant. George Daly, the head of the Harvard team that wrote the review debunking the STAP cells, suggested a plausible explanation. A group of people who were inexperienced in a certain field misinterpreted a result, the green glow, and disregarded contradictory evidence. Essentially, because Obokata and everyone in Vacanti's lab were absolutely convinced that the green glow meant that she had definitely produced pluripotent cells, Obokata might have thought that any evidence to the contrary was wrong, and might have felt it was okay to falsify some experiments supporting pluripotency. At Harvard, Chuck Vacanti took a sabbatical due to the embarrassment caused by the STAP scandal. Despite everything, he remained confident that his core theory about STAP cells was correct, and told a reporter that he would believe in it until his dying day. Vacanti and his lab director Koji Kojima decided to attempt reproducing the STAP experiment to salvage their reputations. They started from scratch, modifying some of Haruko's procedures and using the correct tests to check for pluripotency while avoiding the previous fluorescence mistake. Their plan was to convert skin cells into unspecialized cells using an acid bath, then expose the unspecialized cells to a culture that promoted conversion into nerve tissue, an entirely different cell type. They identified molecular markers which indicated that they had successfully deprogrammed the cells into an unspecialized state. However, they weren't able to coax these unspecialized cells into becoming a new type of cell. A lab in Finland replicated these findings. A scientist in Hong Kong managed to deprogram the cells, but halted his experiment after receiving excessive media attention, fearing the media were trying to use his findings to vindicate Obokata. A surgeon at Walter Reed Medical Center explored a phenomenon observed in soldiers returning from Iraq and Afghanistan with blast wounds from bombs and IEDs. Doctors had noticed that some patients began growing bone in soft tissue where it shouldn't be. The army surgeon, Leon Nesti, tested these unusual sites and discovered they contained 2,000 times more stem cells than expected, and the stem cells seemed to have originated in the local tissue. The surgeon theorized that the blast wounds might have triggered a transformation of cells in the wound area into stem cells, some of which then converted to bone, essentially, the STAP theory that Vacanti had proposed. However, fearing he would be ridiculed by the scientific community if he admitted that he believed STAP cells were real, Nesti simply stated that cells in the area became regenerative and attempted to grow new bone, and avoided mentioning STAP altogether. This situation is reminiscent of the conundrum created by Elizabeth Holmes's Theranos scandal. Some scientists believe that one day, the technology of manipulating microscopic quantities of fluid will advance far enough to run up to 90% of common blood tests on a single drop of blood. However, due to Holmes's actions, many now fear that the entire concept might be treated as a scam, making it difficult for any companies attempting to pursue it. What became of those involved in the STAP scandal? Charles Vacanti's Harvard lab eventually ran out of funding, and shut down. Wakayama's career remained unscathed, and he continued publishing numerous papers on developmental biology and stem cells. The media remained fixated on Obokata for months following the scandal. A video game developer created a character based on her in the game, Star Galaxy, a space simulation MMORPG. The character, Haruko Oppenheimer, was a scientist working on dangerous research to create the ultimate cell. After being criticized for poor taste, the game developers apologized and changed the character's name. A budget retailer sold a cosplay costume called the Rikejo Kappogi, or Science Girl Apron, which came in packaging adorned with flasks, beakers, a notebook, and mathematical formulas. Her scientific career in tatters, Obokata faced humiliating rumors that she would pose nude for a racy magazine, for the equivalent of $350,000, out of desperation for cash, as other Japanese female personalities in similar situations had been known to do. Obokata re-emerged for a magazine interview to promote her new memoir about the STAP scandal, entitled, That Day. She admitted to writing the book because she needed the money. She had reportedly received an advance of $50,000 from the publishers. The book reached #1 on Amazon Japan. In the book, she insisted that she was framed. In the magazine interview, she spoke of vicious male jealousy in the sciences. In 2018, Obokata appeared in a magazine photo spread featured alongside a serialized story called Haruko's Diary, the record of my lonely struggle as I almost killed myself after the STAP incident. Wearing Gucci clothes worth over $1,500, and photographed by one of Japan's top portrait photographers, some members of the public criticized her for a celebrity-like spread that revived memories of the painful scandal, while other outlets speculated that she once again likely needed the money. Subsequently, she disappeared from the public eye yet again. In 2020, several online magazines reported she had been spotted working in a Tokyo cake shop.